Welcome to the third part of Chapter 1. This section, 1.3, is a bit longer, so we're going to divide it into two videos, Part 3a and Part 3b. In Section 1.3, we're going to be looking at different types of microorganisms and their major characteristics, including acellular microbes like viruses and infectious agents. By the end of this section, you'll be able to describe their similarities and differences, and you should also be able to provide a very general overview of the field of microbiology using your knowledge from the entire chapter, Chapter 1. So most microbes are single-celled. They're unicellular and microscopic, so you can't see them without a microscope, but there are some that you can see. And the general rule is in order to be able to see something with a naked eye without a microscope, it has to be about 100 micrometers in diameter. So that symbol refers to micro, or that's equivalent to 0.1 millimeters. So that first little m refers to milli. And I show a ruler down here on purpose. Down here there's an inches. You can measure things in inches or centimeters. So going from, for instance, the number 5 to number 6, that would be 1 centimeter right there. And we also see millimeter markings, which are the tiny lines, and there are 10 of them going from 5 to 6. So that would be 10 millimeters going from 5 to 6. So each of these little lines going from one line to the very next line, and if I were to draw a dot right there, that would be about 1 millimeter in diameter. And of course we can see that with the naked eye, but according to our standard, if something is 1/10 of this, so 1/10 of this, we can still see it. So we probably wouldn't be able to see any details, but we would see something like a little speck with our naked eye. Another standard is shown here. Of course, cells and viruses can differ from these listed numbers, but on average, you might say that an animal cell is about 10 micrometers in diameter. Bacteria can be 10 times smaller, and viruses can be even 10 times smaller than the bacteria. So these we generally cannot see without a microscope. We learned earlier that microbes not only differ in size, but can also differ in their structure, metabolism, and habitat. Some of them are unicellular, but others can be multicellular, and some of them are even acellular or not cellular, such as viruses and a few other infectious agents we'll talk about soon. So for those that are acellular, like viruses, these do not belong to one of the three domains. The others all belong to one of these domains. So other microbes belong in bacterial domain or the archaeal or the eukarya domain. This is a diagram depicting the relative sizes of various microscopic and non-microscopic structures. We can see that from about 100 micrometers and higher, we could see these with a naked eye without a microscope, but of course we can still use our regular light microscope to visualize these items. These include cells, bacterial cells, maybe even some cellular structures, and in some cases the very larger viruses. When we get into the nanometer scale, so N refers to nano, that prefix, we are going to need a much more powerful microscope. And this is usually to see structures inside of the cell and smaller proteins. We can even see things at an atomic level once we get into even more powerful microscopes. And we'll talk about microscopes in Chapter 2. In microbiology, just as we do in other science courses, we use the International System of Units. In chemistry, you might have used liters when measuring volume or grams when measuring mass. For length, which is my example for today, we're going to use meters, which is abbreviated small m. In the international system, we also have prefixes, which are easy ways to show how big or small a unit is by using powers of 10. And prefixes are added before the unit name or symbol. You can see very commonly used ones here on the left, and the prefix is always before the m that stands for meters. Decimeter means 1/10 of a meter, and I can see that depicted here. So 1 decimeter is 1/10 of a meter, And I can also show this by writing that in scientific notation. 10 to the minus 1 is the same thing as 0.1 because I took 1 and I moved the decimal point one space to the left. So 0.1. Another way you can describe decimeter is saying that in 1 meter, you have 10 decimeters. Going through the other units, you can see that 1 centimeter is 1/100th of a meter. A millimeter is 1/1000th of a meter, and so forth. So take a look at this. We want to be familiar with these as we often see these in the laboratory as well. Let's start our overview of microorganisms with our prokaryotes. These are going to include bacteria and archaea. All right, here we have our first prokaryote, bacteria, our main microbe we'll be working with in the lab. Remember that prokaryotes do not have a nucleus. That means their DNA is not membrane bound. It's just sitting in the cell in a region we usually call the nucleoid. So no nucleus. They're found almost everywhere. Remember, they're ubiquitous. So they are also on and inside our bodies. And most of them are harmless or even perhaps beneficial to us, but there are some that are pathogenic that cause disease. They usually have cell walls, and their cell walls are made of a unique substance known as peptidoglycan. We'll talk more about that soon. There are very common shapes found for most microbes, most of these bacteria, including spherical shaped, coccus is singular, cocci is plural. Bacillus is rod-shaped, that's singular. Bacilli is plural. And they can also be curved. So Vibrio kind of looks like this. Spirochete looks like a corkscrew. And Spirillum, this kind of looks wavy. They're very diverse in their metabolism. Some of them are photosynthetic, such as cyanobacteria. Most of them, though, are non-photosynthetic, and they rely on others for their organic and inorganic compounds they need. Of course, because they live almost everywhere, they can survive in a very broad range of environments. Here's another look at those common bacterial shapes. We have spherical cells, the coccus, bacillus, the rod-shaped cell, Vibrio, curved. We have coccobacillus, which is just like what it kind of sounds like, in between sphere and rod. We have Spirillum, which is kind of wavy, and then spirochetes, which look like a corkscrew. Onto our second type of prokaryote, the archaea. Remember, these are unique from bacteria. They're in a different group evolutionarily. These are also single-celled prokaryotes without a nucleus, but they're different in terms of their genetics. Remember that ribosomal RNA sequencing puts them kind of more closely related to eukaryotes than bacteria are. They differ in their metabolic pathways as well as their cell wall and cell membrane composition. They don't have the peptidoglycan that we see in bacterial cell walls. They might have something slightly similar, though, called pseudopeptidoglycan. Okay, so they do not have peptidoglycan. They can often also be found in extreme conditions, including very hot, very cold, acidic, or salty conditions that you might not find other microbes in. And some of them do live on and inside of our bodies, but so far no archaea have been found to cause diseases in humans yet. This is a picture from our book showing the hot springs at Yellowstone National Park, and there are different types of archaea, but also some bacteria in there as well. So towards the center, it's extremely hot, about 70 degrees Celsius or almost 160 degrees Fahrenheit, and that's where you would find these extremophiles, these archaea that can tolerate these extremely hot temperatures. And on the outsides, you might see some other bacteria as well that can tolerate a little bit cooler, but still very hot temperatures. Another checkpoint here, what are the two main types of prokaryotes? Remember, we have bacteria and archaea. Bacteria were single-celled prokaryotes. They had their cell walls that were composed of peptidoglycan, found everywhere, including inside humans. Although most of them are beneficial, there are some disease-causing pathogenic bacteria. There are 3 major shapes: cocci, bacilli, and spirilla, but we saw a few others as well. They were diverse in terms of metabolism. Some of them are photosynthetic, although most are non-photosynthetic. For archaea, also unicellular prokaryotes, but evolutionarily distinct in terms of genetic sequence. Remember that they have cell walls, but those cell walls are not made of peptidoglycan, and they are often found in extreme environments, also found in our bodies and inside our bodies, but not known to cause disease yet. Now we're going to switch gears and look at eukaryotic microorganisms, and these will include things like algae. We've got some protozoa here, some fungi here and here, and then helminths, which are parasitic worms. The domain eukarya includes all eukaryotes. Remember, these are cells with a nucleus, meaning their DNA is bound with a double membrane. This includes protists, fungi, plants, and animals. Protists are pretty diverse. So things that are not protists include plants, animals, or fungi. Instead, protists include algae and protozoa. Protists can be unicellular or multicellular. And algae are mostly protists, and they're very diverse. They change or they vary in size. We're going to see many different types of structure and habitat in the future. Most algae have cellulose in their cell walls. So their cell walls are composed of cellulose, and most also perform photosynthesis. That means they're able to capture carbon dioxide from the air, fix that, and produce oxygen and carbohydrates for energy. You have to be careful. Cyanobacteria sometimes is referred to as algae because in the past, when they first discovered these photosynthetic bacteria, they thought they were algae, but they are actually not. Cyanobacteria are photosynthetic bacteria, so these are prokaryotes. Similarly, as you read through Chapter 1, specifically Section 1.3, and you get to the protist section, you'll see the sentence, some algae are protists and others are bacteria. Let's cross that out. That's an error. Remember that while the term blue-green algae was previously used to describe cyanobacteria, now we know cyanobacteria are bacteria and not algae. Algae are eukaryotes. Bacteria, like cyanobacteria, are prokaryotes, so in very different domains. Our first eukaryote is algae. So algae plays key roles in the ecosystem because they go through photosynthesis. They can support other organisms by undergoing photosynthesis, capturing carbon from the air and producing carbohydrates, these energy-rich byproducts. We also see a lot of algae in our daily consumer products. So carrageenan and alginic acid, these are really good thickening agents. So you'll see them in foods like ice cream. You'll see them in cosmetics and toothpaste. Agar is also from algae, and we're going to see this a lot in our lab, in our petri dishes, and in our test tubes. Algae is also well studied as a biofuel source because this is a type of renewable energy source. And then at the bottom, I put other examples where you might have encountered algae. The kelp that you see in the ocean, brown algae. You might have eaten red algae or nori, and then here's some seaweed salad on the left. Diatoms are another type of algae. They're found in oceans, the soil, and they're interesting because they're surrounded by a cell wall made of silica. So they're sometimes called the jewels of the sea because of their appearance. And if you've ever used diatomaceous earth at home, diatomaceous earth is made from diatoms. So this is useful for insect killer here basically, but you can also use it to dry things out for filtration or insulation. Our second eukaryote are protozoa. So just like algae, these are also protists. And these protozoa serve as the base of many food webs. So they are the food for many other organisms. They're very diverse as well in terms of form and function, and most of them live in water. If they live in water, then they have to have some kind of structure to allow them to move around. So these can include cilia. So cilia you've probably seen before in other classes, little hair-like proteins that help the cell move around or the cell move things across its surface. Flagella, which are usually longer proteins, and you can have one, two, or several on the cell surface, and pseudopods, which are extensions of the cell membrane, and we'll see these in amoeba later on. For protozoa, they can be photosynthetic or rely on other organisms for food. They can be free-living, so they can live on their own, or they can be parasitic, where they depend and live on or inside a host for nutrients. Most of them are harmless, but some can be pathogenic and cause disease. Our textbook gives us an example of Giardia as a type of protozoa. So I can see several flagella here for motility, for movement, and then this is something called an adhesive disc. That is what Giardia lumblia uses to attach to our GI tract. And one of the signs you'll see for people who are infected is severe diarrhea. Our third eukaryote are fungi. Remember, fungi are not protists. Protists were the algae and the protozoa we were talking about earlier. Fungi are eukaryotes, but they're not plants because they cannot go through photosynthesis. Instead, they get nutrients by absorbing organic matter from other organisms. Unlike plants, their cell walls contain chitin, whereas plants had cellulose cell walls. And they're found in many places as well. We can see them in the soil, they're in water, in food, and on our skin and inside our bodies as well. Down here I put examples that we might have encountered in other courses. We have Amanita, which are a popular mushroom because we see them in like Super Mario, in different books. I have a necklace that looks like this. And then on the right is an interesting one called Xylaria, also known as dead man's fingers. This is an interesting fungus that looks like fingers, right, coming out of the ground. So fungi are a type of kingdom with several forms, but the main types include yeasts and molds. Yeasts are single-celled fungi that kind of look like an egg-shaped structure. They're often used in baking and fermentation and found in many different places, like the ocean to the human skin. They can cause spoilage of our foods. They can also cause infections, such as different types of skin infections or thrush, which is kind of a white growth in the back of the throat, or vaginal yeast infections. Molds look different from yeasts. They're made of filaments and they're multicellular and they look like string-like structures. I'll show you some pictures in the future, but in a bit. Like that, except my marker's too thick. They're good at breaking down dead organisms and recycling those nutrients, but they can cause allergies in some folks, and they can also, some of them, produce harmful toxins known as mycotoxins, with the prefix myco referring to fungi. Some of them can be used to produce medicines like the antibiotic penicillin or the immunosuppressant cyclosporine. Here's a picture of yeast, specifically Candida albicans, which is a type of yeast that can cause thrush or vaginal yeast infections. Looking at the picture, you can see that these are single-celled structures here, and they're kind of an oval shape. And in contrast, here's mold on the top of these moldy oranges, and if you zoom in, you can see that they're hair-like filament structures. Here I've put the two side by side. We've got yeast with their unicellular egg-like shape, and on the right we have mold, which are these string-like filamentous structures. Another checkpoint. Let's go through this briefly. Make sure you guys go through it in more detail. Two types of protists. We talked about algae and protozoa. Two types of fungi. We talked about yeasts and molds. And name some of the defining characteristics. So algae, we said they're photosynthetic. They can be uni or multicellular. They had cell walls made of cellulose. Protozoa, they usually live in water, and they're also unicellular. Because they live in water, most of them, they either have cilia, flagella, or pseudopodia, the pseudopods, those false feet. Some of them can be photosynthetic, others depend on other organisms for nutrients. And we talked about fungi. We have yeasts that are single-celled. They can cause things like thrush or yeast infections. We had molds that are multicellular, remember, made of those filaments. They feed on dead organisms. They can cause allergies or produce harmful toxins, but they can also make medicines for us. All right, so that's the end of our first video for section 1.3, and the next video will continue with our eukaryotes, specifically helminths or parasitic worms.